Welcome back to the Answer Series Diagnostic Report teaching and learning tool for the NSC November examination. This is an all-in-one tool that combines the November NSC paper, memoranda, and annual diagnostic report commentary to create a comprehensive learning experience. TAS provides this document for free, and we encourage teachers, learners, tutors, and subject advisors to use it. All we ask is that you please give credit where credit is due and not change or alter any content in this document that removes our name from it. Continuing with Section A's multiple choice, the first slide in the teaching document will provide the question, the possible answers, as well as the page references for our Grade 12 Life Sciences Part 2 edition. The second page provides the answers. Let's run through them. Which one of the following organelles is the site for protein synthesis? The answer there is A, ribosomes. In humans, the gonosomes determine the gender, which combination below shows the correct gonosomes for males and females. The answer there is C, males have gonosomes XY and females have gonosomes XX. An organism has the genotype big T, big T. The correct term which describes the organism's genotype is homozygous dominant. Heterozygous means that the two alleles are different. In this case, they are both a big T, so that means the only options were C and D. The fact that they are both capital letter T's indicates that these are dominant alleles, so this genotype is homozygous dominant. Down syndrome is a genetic disorder where an individual has an extra copy of chromosome 21, which one of the following may lead to Down syndrome. As Down syndrome is caused by an extra copy, it means that either the sperm cell or the egg cell was carrying an extra copy during fertilization, which led to 3 copies in the final somatic cells. So this is a problem that occurs during meiosis, and therefore option A concerning mitosis should be disregarded. That leaves us with B, C, and D. Option C talks about chromosomes failing to form pairs during fertilization, which has nothing to do with the availability of an extra chromosome, so that removes C. And then B and D both concern meiosis. B is the correct answer, which concerns the separation of chromosomes that does not occur correctly during meiosis, which basically means that in the case of Down syndrome, possibly both homologous chromosomes move to one pole. They didn't separate during anaphase 1, or in the case of anaphase 2, the chromatids did not separate, leading to one gamete having an additional copy. D is not an option because it talks about crossing over, whereas crossing over has to do with the exchange of genetic material and not necessarily the separation of chromosomes or, in fact, the doubling of a whole chromosome in total. So the only correct option there was B. A gradual change in the characteristics of a species over time is known as D, biological evolution. Candidates saw the word gradual in the question and then just assumed that this was referring to punctuated equilibrium. This may be because learners confused the concept of punctuated equilibrium with gradualism, which is often used as a comparison with it, where gradualism is the slow, gradual change of a species in time, which is the typical idea of Darwin's evolution, whereas punctuated equilibrium actually explains the speed at which evolution occurs. It says that there's long periods of no change or stasis in which species remained unchanged, and then during this it's punctuated with short periods of rapid changes. So this definition refers to the overall definition of biological evolution, the gradual change in the characteristics of species over time, which is also typical of Darwin's idea of evolution. Then questions 6 to 9. Which one of the following is a reproductive isolation mechanism? So learners merely had to recognize the ones that they've learned. Adaptation to the same pollinator should be adaptation to different pollinators. C is incorrect. Production of fertile offspring should be production of infertile offspring. Breeding at the same time of the year, number D, is also not correct because it should be breeding at different times of the year. So the only one that reads exactly as the options they learned is B, species-specific courtship behavior. Then analysis of mitochondrial DNA is an example of this line of evidence. Fossil evidence does not have anything to do with mitochondrial DNA. Modification by descent also links to structures and not necessarily DNA. Biogeography has to do with the location of living organisms on Earth. So the only one that actually deals with genetic evidence is D, genetic evidence. And DNA is obviously a genetic evidence. Which option in the table below shows the correct comparison between mitosis and meiosis? So candidates confused the processes and products of mitosis and meiosis, and in video 1 of this paper's series, we provided an overview diagram to show the differences between these two processes. So the correct option here was C, that mitosis, the number of chromosomes will remain the same, and in meiosis, the number of chromosomes is halved. The only way to reach that conclusion is to go through all the other options and to go through a process of elimination. So A, mitosis did not produce 4 daughter cells, it produces 2, which is in this case written at meiosis, so those 2 needed to be swapped. B is incorrect because mitosis does not produce genetically different cells. That is the process of meiosis. And again, the 2 options are swapped. And then D, 2 divisions occur in mitosis is also incorrect. That is a characteristic of meiosis, whereas 4 divisions occur in meiosis is not a characteristic of either one. So therefore, A, B, and D are options that you can eliminate based on their content. So C is the only remaining correct option. Which one of the following scientists discovered the fossil the tongue child? The answer there is C, Raymond Dart. Candidates did not know the name of the scientist. This is one of those things that they unfortunately need to memorize, which is difficult. There are many different memorization techniques or teaching tips that we could go through, but we've added one example of such a memorization technique in the Let's Revise This page. So here's a table showing us all the important fossils and the paleontologists that discovered them. Now obviously this is a lot of scientific terminology to memorize and also remembering names of obscure people, which learners often find difficult. So an example of a memorization technique could be that instead of trying to memorize them straight out, who discovered which fossil, try to make up a story using the names. And hopefully this sparks the full terms when you remember the story. And this is a silly little example of that. So Ardi's fossil was very white. So Ardi was discovered by Tim White. Whereas Lucy's looked like Donald Duck. So Lucy's fossil was discovered by Donald Johanson. She visited Laetoli and left many leaky footprints. So the Laetoli footprints were discovered by the Leakey team, Mary Leakey's team. All over Mrs. Pless's floor, and she was using the broom to sweep them up. So Mrs. Pless was discovered by Robert Bloom. When the tongue child darted past, so the tongue child was discovered by Raymond Dart, leaving a little foot imprint. Her brother Ron, so Littlefoot, discovered by Ron Clark, shouted "Karabu" and made a Bruce Lee karate chop. So Karabu, discovered by Lee Berger. So you can try to modify a story like this to get the names of the fossil and the person in the same sentence. My idea was basically just making sure that they're in the correct sequence. So learner could go name and name. The first 2 names go together, the second 2 names, the third 2 names. So in that way, hopefully it creates a little picture of the fossils and who discovered them. So that's just a tip on an exam and memorization technique. So returning home, we look at the last multiple choice question 1.110. Which one of the following is produced at the end of translation? So that's the whole process of protein synthesis ends with translation, so it only makes sense that the one thing followed at the end is a protein. Then terminology, question 1.2. Learners are instructed to give a biological term for each of the following descriptions. Important things to remember when it comes to terminology is to make sure that you're providing a term and not an acronym, and that you're providing a single term and not multiple terms or phrases, because the marker is instructed to provide a mark for only the first thing that is provided as an answer. So question 121 says a nucleic acid that carries hereditary information, that is a deoxyribonucleic acid. In this instance, they did accept DNA as an accredited abbreviation. Learners, however, did not provide the correct abbreviation. They might have provided RNA as the nucleic acid that they often confuse DNA with. Number 2 is the chromosome condition of a cell that has a single set of chromosomes. So the answer there is haploid. A lot of learners read the word condition, and this created confusion because it made learners think that there was something wrong. There might have been a disorder. So that was a problem with the wording of the question. So learners in this regard often provided the incorrect answer, aneuploidy, which is also not actually mentioned as part of the exam guideline. Learners do not need to know about aneuploidy. Question 3, the structure that holds the two chromatids of a chromosome together is that middle section, the centromere. And here learners provided a whole range of terms that they often confuse centromere with, and we went through the example of how to distinguish between these in the previous video. So they provided centriole, centrosome, or chromosome instead of the correct centromere. Question 4, the type of RNA that carries specific amino acids to the site of protein synthesis is tRNA or transfer RNA, and again learners did not provide the correct abbreviation. They might have written it with a capital tRNA, as was indicated in the general comment section about correct spelling. Number 5, the analysis of DNA samples that identify individuals or relationships between individuals. So it's the analysis, it's the process that's involved here. So that's DNA profiling. It's not a DNA profile. That is the product. That is the thing that will be analyzed. So the analysis process, the creation of a DNA profile, is DNA profiling. Question 6, a representation of the number and structure of all the chromosomes that occur in the nucleus of a somatic cell is called a karyotype. In this exam, karyogram was also accepted. Question 7, a group of similar organisms that are able to interbreed to produce fertile offspring is the definition of a species. This was confused with the definition of a population, and in the previous video, we went through an example of how to explain the difference between these two terms. Question 8, the phase of meiosis where paired chromosomes are arranged at the equator. So it's a double row, it's a row of paired chromosomes. So that's metaphase 1. And very, very, very important when it comes to meiosis is you can never write the name of a phase without indicating a 1 or a 2. So in this instance, learners lost marks because they failed to indicate the I or the 1 as part of their answer. They just provided metaphase. And then lastly, the biotechnological process that produces genetically identical organisms is cloning. Question 1.3, the items and statements, where learners had to determine if A only, B only, both A and B, or neither of the two options applied to the statement. A source of variation in populations. Options provided include random fertilization and random mating. Both of these lead to variation within populations. Question 2, the discovery of the structure of the DNA molecule is only A, Franklin and Wilkins. Lamarck and Darwin are scientists involved in evolution theory. So the diagnostic report says that candidates struggled with the history of DNA's discovery. Question 3 occurs during meiosis, is both karyokinesis and cytokinesis, as we looked at in the previous video on the general comments. Candidates chose the incorrect option as they were most likely not familiar with the word karyokinesis in cell division. So it's important to unpack that term for learners, that karyo means kernel in Latin and kinesis means to split. So karyokinesis is the division of the nucleus. Question 1.4, the diagram represents a DNA molecule undergoing an important biological process. The only biological processes that learners are taught that DNA is involved in is either DNA replication or protein synthesis. And in that sense, it's only transcription, the first half. So looking at the diagram, the first thing you need to do is figure out which biological process is actually being shown. That's also the first question. And the answer is DNA replication, because we can see that it's a single DNA molecule that's making a copy of itself. There is no mRNA codons coming in, no other nucleotides, no single-stranded structure is available that could represent mRNA. We can actually see a double helix that has split, and that is being produced, a copy of it is being produced on either of these two strands. So you're actually reading the diagram from the bottom upwards, where the split occurred in the original DNA strand and new DNA strands are being compiled on either side. The chemical bond number 1 is called a hydrogen bond. That keeps the nucleotides together. Question 1.4.2 asks to give the collective name for the parts labeled 2, 3, and 4. Now, the diagnostic report comments on the fact that candidates misread and provided the names of the individual components 2, 3, and 4 instead of the collective name as it was asked for. A better way to possibly have asked this question is to rather have added a little block around that section rather than giving them individual numbers, because this does contribute to the problem the diagnostic report mentions, especially for third language speakers. So they wanted the answer nucleotide, which refers to that whole structure consisting of a phosphate group, a deoxyribose sugar, and some other nitrogenous base represented by number 4. Then they also asked for the full name of the nitrogenous base labeled number 5. So in order to determine that, learners had to read the diagram from the bottom up. So G represents guanine. That would have connected with the nitrogenous base on the other side, which would have been C. Then the A is the second nitrogenous base shown, and that connects to the unknown base number 5. So the complementary nitrogenous base for A is thymine, and that needed to be spelled out because the question specifically asks for the full name. Question 143 says, during which phase of the cell cycle does this process take place? That should also have hinted to learners that the answer for question 141A is DNA replication, as DNA replication is the only one that occurs during the actual cell cycle. Protein synthesis, specifically the transcription section of it or part of it, in which DNA is directly involved, doesn't necessarily occur within any specific area of the cell cycle. It's not like mitosis or meiosis is essential for that process to take place. So that should have hinted to the fact that this is still DNA replication. The answer obviously being interphase. Some candidates identified the phase as interphase 1 or interphase 2. However, interphase does not occur separately in meiosis 1 and meiosis 2. So it's the only one of the phases that you don't need to add a 1 or a 2. Very ironic that they are adding it here when it's not necessary, but then in question 1.2, where it is necessary, they leave it out. Question 144, where in the cell does this process occur, meaning DNA replication? It occurs within the nucleus. Question 1.5 has to do with genetics. In a certain species of butterfly, one gene controls wing spot color and another controls eye color. So we have 2 different genes, 2 different characteristics. So immediately you feel dihybrid cross. The wing spot color can be red or gray, and we can see from the annotation provided that red spots are the dominant traits, gray spots is the recessive trait, while eye color can be black or brown. Black here with a capital E designates the dominant trait, and brown with the small letter e designates the recessive trait. Butterflies that are heterozygous for both genes were crossed. So that means that butterflies that were crossed together had this genotype combination. They were heterozygous for both genes, in other words, the gene for wing spot color and the gene for eye color. Name the type of crust represented above, as there are 2 different genes controlling 2 different traits. This is a dihybrid cross. Question 152 asks to give the dominant characteristic of each gene. In order to determine that, you just have to look at the annotation that the question provides. The capital letters represent the dominant genes. So the capital R means that red spots is a dominant characteristic, and the capital E means that black is the dominant eye color. The diagnostic report says that some candidates listed the dominant alleles, so they went big R and big E instead of the dominant characteristic. Some candidates only provided the colors instead of the full description of the characteristic, so they just wrote red and black instead of what is red, the red spots and the black eye color. And this is really important to teach learners to write full words, full phrases, full sentences, because a characteristic is not just red or black. Different parts of the butterfly is red or black. It has red spots and black eyes. Continuing with question 153, it says that the table below shows the phenotypes of all the possible genotypes of the offspring. So the phenotypes are the physical characteristics. They show them red spots and brown eyes. They give you an unknown phenotype Y. They say gray spots and brown eyes, and then they provide you with the possible genotypes where you have to provide X. They give you the genotype for the unknown phenotype, big R, big E, small e, and then gray spots and brown eyes is the completely recessive genotype with 2 small r's and 2 small e's. Then they ask you for the possible genotypes at X, and they ask for all the possible genotypes. Now remember that red spots is the dominant trait, and brown eyes is a recessive trait. So the easiest thing is to always start with a recessive trait, because with a recessive trait, the genotype is known. It can only consist of 2 recessive alleles, small e. To have red spots means that it has at least 1 dominant allele for the color of the wing spots. So it has to have 1 big R. And then the butterfly could have a second big R, or it could have a small r. So either the butterfly is homozygous for wing color or heterozygous for wing color. In either case, the red being the dominant trait will be expressed. The diagnostic report says that some candidates lost mark for the incorrect presentation of the alleles. They separated the gene alleles from one another, so they wrote RE instead of keeping the genes together. So R's go together and E's go together. First you write the R's, then you write the E's. So stick to the annotation as provided in the question. And also learners need to note that they gave them the answer in terms of how to write it in the table, because the table shows them that they keep the R's and the E's together. Then question B says to give the phenotype at Y. So they've provided you with a genotype. It's a big R, big E, small e, which means that the phenotype of this organism for wing spot color will be big R, meaning red spots. And then eye color is a big E, small e combo, which means the big E that codes for black eyes will dominate the brown eyes. So characteristics that you can see of this butterfly will be that it has red spot and black eyes. And then the last question says the genotype of the gametes produced by a butterfly with gray spots and brown eyes. So you can look at the table, gray spots and brown eyes, the genotype there has been provided. Remember that when there are 4 letters together, meaning 2 different characteristics and their alleles, this is of a somatic cell. The question is asking for the gametes. And during gamete formation, gametogenesis or meiosis, the alleles have to separate. So if there are 4 letters in the somatic cell, then the gamete has to have only 2, 1 of each. Now this is easy to determine if you use the FOIL method as set out in our grade 12 Life Sciences Part 2 study guide, where you basically do first letters together, outer letters together, inner letters together, and last letters together to give you 4 possible gametes that are produced by the genotype of this organism. So the correct answer here is a small r and a small e. That's the only possible genotype for the gametes. Candidates were unable to formulate the genotype of the gametes, and instead they gave the genotypes of a somatic cell, where they basically just copied what was in the table with the 2 R's and the 2 E's. So this is definitely something learners do struggle with, and we've provided some revision in this regard. So this is a similar table to the one that we used in the previous video, where we looked at the difference between monohybrid and dihybrid crosses as commonly confused processes for learners. You can return to that video to revise that. So in this table, just once again, all we've added here is that the genotype notation and the gamete notation should be considered, and they should be noted that they're different. So in terms of the genotype notation, this which is provided in the table shows a somatic cell, a diploid cell, where you have 1 allele from each parent for each of the characteristics involved. So green could be big G, where one of the big G's comes from the mom and the other one comes from the dad, maternal and paternal chromosomes. Then big G, small g, one comes from the maternal chromosome, one from the paternal chromosome. So the point is, a genotypic notation of a somatic cell for a monohybrid cross consists of 2 letters. Then if we look at a dihybrid cross, the somatic cell has 2 of each letter, and the letters stay grouped together because they are showing you 2 characteristics. So you have a big G, one from mom and one from dad, maternal and paternal, and then a different characteristic will also be concerned, namely in this case the wrinkled or smooth, the seed texture, and the big R, one of them will come from the paternal chromosome and one from the maternal chromosome. And that's true for all the different genotypic notations. When we look at the gamete notation, in a monohybrid cross, as there's only one characteristic involved, a sperm cell or an egg cell will only carry one example of that specific trait because it's receiving half of the genetic information. So only a big G or only a small g. But when we look at the gametic cells within a dihybrid cross, they will carry 2 letters because it's a letter per characteristic. That sperm cell that's produced during spermatogenesis will carry a allele for seed color, and that could be either green or yellow. It will also carry an allele for seed texture, which could be wrinkled or smooth. So note that there are 2 letters, one of each. Then returning home, that concludes question 1.5, and we move on to the suggestions for improvement for section A. So the following issues were reported in the Diagnostic Report of 2023, but it continues to be a challenge, namely the steps in DNA replication, the genotypes of gametes in a dihybrid cross and how to write them, and the name of scientists and the fossils that they discovered. And many of these were revised in our teaching and learning tool from that specific paper, so go and have a look at that on our website.
If you found this video useful, give it a like and subscribe to our channel so you don't miss any new episodes. Follow us on Instagram or Facebook to stay on top of the latest TAS news and launches. So that's it for now from the Answer Series, your key to exam success.